Well, thank you for having me. Yeah, so my name, well, let me make sure I can. Yeah, my name is Alonso Delgado, and I am a evolutionary marine biologist by training. And I am a fourth year PhD candidate at Ohio State, and that is in Columbus, Ohio, right in the middle of the country, with there's no sea or anything around us, which is great. And so I work for Dr. Meg Daly, and we work within the Museum of Biological Diversity on campus. My research focus has two major tracks. One of them is sea anemone taxonomic diversity. So we really look at species level relationships, whether that's using PopGen, phylogenetics, species descriptions, and then into that we've been incorporating venom studies to understand differences in venom composition across many of these different species and doing some case studies in phylogenetic scaled approaches, at least for some of the future work that we're doing. And we're also really interested in understanding quantitatively how to quantitate venom diversity, alpha and beta diversity in sea anemones. What does that look like when we look at what drives these trends across large evolutionary scales? But today we're going to be, like I said, we're focusing on venom, and we're focusing on venom because sea anemones are very interesting from an evolutionary perspective. However, everything or most of the things we know from venoms has been inferred from three key main groups. They're model organisms for venoms by any stretch. And we focus on these groups, and there's a lot of phylogenetic thinking and large-scale studies and well-scaled numbers of studies that look at both venom at a macro and a micro level, so at the individual level and at a phylogenetic level. What we find when we're looking at these groups and we're thinking evolution for these individuals, we find that their venoms evolve under positive selection. So their venoms are evolving to diversify and potentially maintain an evolutionary arms race with their prey. One key feature that these group of organisms share is that they're evolutionarily young. So all within 100 million years, give or take a couple million years. However, when we think about the oldest evolving evolutionary group, phylum, that has every single one of its members being venomous, cnidarians, we find some very cool distinctions when we look closer. So the most well-studied group within cnidarians is sea anemones. And when we look at sea anemone venom, we find that sea anemone venom evolves largely under negative selection, which is a contradicting statement relative to the other well-studied groups when we're looking at modes of evolution, at least. So because of that, we really have to ask, why is evolution acting differently on a convergent trait? They're convergent both at a functional level and a compositional level, at a transcript and a proteomic level. But our thinking behind this is that it is an age component to this question. That if we're basing a lot of our analysis at what we know from snakes, and we're using snake references as our key, at least, there's more snake transcripts when we're looking at transcriptional comparisons that we're potentially inferring things evolutionarily wrong, because these comparisons that work with snakes and all the other lineages might not scale up phylogenetically. And we're talking about sea anemones, which are closer to, or at least cnidarians, which are closer to 800 million years old. One of the key things that I like to focus on or think about is that within sea anemone studies that come to this assumption of them evolving largely under negative selection, is there's a low number of species representation. There's a low representation of the types of venoms that are looked at, and these studies often lack a phylogenetic perspective. And in doing so, they use broad evolutionary comparisons that might not be evolutionary sound. So what I mean by that is that some of these studies use species who last shared a common ancestor roughly 500 million years ago, which is older than the last common ancestor shared by all vertebrates. And we wouldn't do one-to-one comparisons with many individuals in this group and make laws that govern, which are meant to govern how we view venom evolution in sea anemones. Another thing that sea anemone venom studies historically have focused on is that they're focused on sea anemone specific venom families, which are neurotoxically dominated. There's a lot of need to do this because a lot of neurotoxin families in sea anemones are now well studied, but they also have biomedical relevance. So there's a large amount of focus in these groups. However, when we published a paper in 22, we looked at the venom composition of a group of ecologically similar sea anemones, so clownfish hosting sea anemones. We found that neurotoxins, at least for this group, only encompassed roughly 30% of total venoms found in their venom. And actually, their venom was dominated by hemostatic and hemorrhagic toxins and venoms. Recently, this other paper came out that using proteomics, that you get the same findings, so at least some validation in that sense too, that there is a difference across ecological groups, perhaps. So given that we have made large assumptions on many levels to try to understand sea anemone venom, I think there's a need to examine venom at both micro and macro scales evolutionarily. And today, the main focus of what I'm going to be talking about is really looking at, do non-neurotoxin venom families for sea anemones evolve differently, i.e. not negative selection at a macro level? So we did this by looking at a set of families, but we're using a case study today that we're going to focus only on one group of insulin-like venoms in sea anemones, and we're calling these guys nidoinsulins. This is part of a paper that I just published, and if you're interested, you're able to go read it. There's a lot more that I'm not going to cover from this paper, that particularly in venom VP32, that there's really cool evolutionary things going on. So what we did, we did very standard transcriptome mining and genome mining. For transcriptomes, we did a pipeline into which we look at protein families, which then filtered for keywords like insulin, and then we annotated those transcripts to get a better understanding of what's there. We created alignments, we created phylogenetic trees, and then the combination of both was fed into DataMonkey to make assumptions on their mode of evolution. So what we found for nidoinsulins is that nidoinsulins have at least two chains, and chain A relative to chain B has a high level of conservation. And chain B has a level of ambiguity across the transcript itself, and at least when you're looking at the alignment, that makes you wonder what aspects of it are, why is this, right? When we create a phylogenetic tree, we see clearly that we find different clades. So some deep-rooted duplication events that have happened in nidoinsulins. And so we find at least 4 clades for nidoinsulins that represent 4 distinctly evolutionarily evolving versions of nidoinsulins. We were very interested in seeing how the diversity in transcript and phylogenetic evolutionary history inferred its function. So one of the ways we thought we would address this is by looking at protein structure. So we looked at protein structure within the framework of a few key organisms in this group for each clade. And we found something quite interesting, but I think it's conclusive to say that when we use the best model chosen for each transcript came from Swiss model, we got a very standard shape, which is what you see on the left side, on the right aspect of that. And we use the AlphaFold, when selected an AlphaFold model to create the protein structure, it showed generally the same, except that the barrel at the very top folds outward rather than inwards. Not all to say is that regardless on the method used, you get consistent structure. So we're making the assumption that the structure is consistent across our studies, and we are looking at the same thing, not looking at one venom that's evolved drastically different as potentially gain new function at least. This was done by one of my colleagues, Kyle Zuzanski. He is also part of the Ohio State group. So one of our things that we found really interesting was that the common way to look at evolutionary rates is to look at the global rate of evolution of any venom family. But when you do that, it's clear that those rates are not representative of what's going on in the clades. So just from that, we can make a quick definite statement that global D&DS rates are not representative of evolutionary rates of each lineage. So we should be very cautious when doing that because it's a very common thing we see in the literature. That actually, when you plot out these evolutionary rates across states, we still see that it's supportive of our phylogeny, that each independent clade is evolving drastically differently and at some points at different levels of magnitude. Which is very cool as an evolutionary finding, but also very interesting because it's maintaining its structural shape when doing so for hundreds of millions of years. So what we can say from this is that negative selection is probably the most dominating force in sea anemone venom coding gene families, but positive selection still happens. It's just not the dominating force. So when we look at sites under positive selection, when doing this analysis, we find that there's also distinction across the clades. So we thought it would be interesting to, when we plot these results out onto themselves, onto each clade, we see that at different points in the clade, there is a lack of representation of a site under positive selection for within clade A1 and A and 1B. What that looks like is that you have it when you look at them both together and in the sister clade at clade 2, but you don't have it at these key points at the tips of the tree, but you have them at the crown groups. When you think about this in a phylogenetic perspective and you plot it out on the tree, we actually see that if it's a really nice evolutionary picture at that, but that we can potentially now find sites that have been possibly under positive selection at some point, and then that signature has been lost. Which means we're probably undercounting positive selection as a whole evolutionarily when we do these kind of studies, because a lot of the times we're not taking phylogenetic perspective, at least in my groups. All to say that venoms of all types of sea anemones are basically dominated by phylogenetic duplication events and deep homology. We find this in all the other venoms we looked at as well. And negative selection, just really, is the dominant evolutionary force. But there is a lack of methods that accounts for those sites that have been lost, and therefore we're getting a skewed view of positive selection within these systems that are dominated by negative selection, at least. And that we're able to actually just find some of these sites, which is really interesting because we could zoom in and key evolutionary, we don't know evolutionary process happened, but we can start to think about what might be going on at these key sites. So just to plug in some future work is that we're looking at how to quantify alpha and beta diversity in sea anemones at large scales, and looking at the drivers that drive any patterns that we find. And some of that work I'll be presenting at the Gordon Research Conference this late summer. So just to acknowledge people that have helped me, some funding sources, and I am open to any and all questions. Thank you for listening.
Thank you very much. Thank you very much, Alonso. So are there any questions? Are there any questions? I have maybe to open the chat. So while we are waiting for the first question, I may have a short one, a more general about the neurotoxins.
Yeah.
And I think it's the most common question that the people might have when you talk about sea anemones. How is the resistance in clownfishes going?
So there's a lot of work that is currently going into this. And I think one of the papers that touches on this more than any other papers is a paper I shared by Casey Hoffner. I would say, in short, we're not completely sure, but there seems to be some secondary effects by the mucus the sea anemones secrete and the fish secrete that might have an effect on any types of venoms binding to receptor sites in fish, which is as close as we're getting to right now. But I would still say we're not completely sure if that's the only reason or the only thing that is really dominating those interactions. Because sea anemones have symbiotic relationships with like many organisms: sponges, clownfish, other fish, pistol shrimps, cleaner shrimp, gastropods, snails, hermit crabs. So there might be some evolutionary umbrella that allows sea anemones to have these relationships, but necessarily with clownfish, that's as close as we're getting. The mucus.
Thank you for the answer. I don't see any further questions, so I will ask the next one. You showed the quantification of the different sea anemones. How were they quantified? Was it protein level or was it RNA level?
So what part were you talking about? So when we looked at
where you showed that there are some neurotoxins, but also some
so
neurotoxins
so that was at the transcript level. And then what I mentioned was Casey's paper did that at the protein level, so using proteomics, and the results were similar. I mean, at least at the functional level, with different types of transcripts being matched, but one paper 2 years later, you get a different type of data set involved. But functionally, when we look at function of the venoms that were looked at, it's scaled the same, or at least very closely.
So the effect of a sea anemone envenomation is likely various as you see it from spiders and from snakes. So they can affect the neural system that is normally common, but they also can affect the blood coagulation or other parts. Okay.
I would hate to ever find a human-sized sea anemone because they will kill you, they will melt you, and they would basically destroy you in every way you could think.
Sounds
we're
like
very
a
lucky.
lovely
They're
object
tiny.
to study.
They're very fun.
And I have a last question. You showed in the overview about the different component in the sea anemone venom, also BBPs. Which size do the bradykinin potential peptides have in the sea anemones?
Like we're talking maximum size?
In general, is it more 5, 4, 3 amino acids or more 20, 30 amino acids?
It's so variable, but I think
Okay.
I don't think I could even put a number on it, but I think their normal baseline we put is that we look at 20 and above, and then we get anything from 400 and to 10. It's so variable, and I think it's part of the issue with sea anemone venoms that we don't know enough. So at least my work is trying to shine some light on that, because our PLA2 study that we did showed many types of PLA2s at many sizes, from 30 amino acids to 200 amino acids. So it's variable. There's no structure as far as we know at this point that dominates how big or how many at any given point.